first things that comes to mind, especially when we think of curing disease, is the use of antibiotics. However, unfortunately, the effectiveness of antibiotics is not going to last for much longer. It is estimated by the year, the World Health Organization has declared antibiotic-resistant bacteria as one of the global threats, and it is estimated that by the year of 2050, 10 million people will die annually because of antibiotic-resistant bacteria. That is 10 million people every year. For drug companies, it's just not worth it to try and develop new antibiotics. It's too complex and it's not profitable enough. That's where we come in, with our alternative, pyacins. So what are pyacins? Pyacins are bacteriocins produced in nature by the bacteria Pseudomonas aeruginosa. They're protein complexes with this structure. It has a sheath-like structure in the middle, a base plate, a core, and numerous tails. Kind of resembles some 5 phages, but it lacked the DNA. We'll talk about it later. And it's basically a weapon. So the way it attacks is that the tails, the highly specific tails, attach to the cell envelope of the rival bacteria. The sheath-like structure contracts, the core perforates the rival bacterial membrane, thus dissipating the membrane potential of the rival bacteria and killing it. It is important to emphasize that engineering pyacins is very new and barely researched. So what we can see on the left is a pluck assay experiment we performed with wild-type Pseudomonas aeruginosa and pyacins. We caused an SOS response, DNA damage in the Pseudomonas, creating the pyacins, 3 drops, where you can see the P on the plate, and we can see that there was no bacterial growth on the plate. So what happens in nature? We have our Pseudomonas aeruginosa. It senses a rival bacteria. It causes an SOS response in the bacteria, the Pseudomonas, mainly DNA damage, that starts the production of pyacin, many pyacins, until we reach about 200. The bacteria explodes, releasing the pyacins. Pyacins attach to the rival bacteria and kill it.
Thank you, William. When thinking about turning this amazing protein into a drug, we understood that 2 things must be done. The first is to change the target of the pyacin, and that can be done by altering the tails. And the second is to change the vessel. That's because Pseudomonas is a really harmful bacteria, so we decided to work with a much friendlier E. coli. So when considering using E. coli, we understand that there is much more potential in it, because we don't only have E. coli in our lab, we also have E. coli in our body. So why not, instead of producing those pyacins only in the lab, let's produce them also in E. coli for our body. And by that, this E. coli will sense the bacteria near it, produce the pyacin. Moreover, we can give it a few tails genes, and by that, we'll be able to create different kinds of pyacins according to the bacteria near it. So, how do we imagine this vision? This is our in-body E. coli drug factory. It contains a tails catalog, which is practically genes of different tails. It can create different kinds of pyacins, different kinds of tails, according to the different kind of bacteria that you meet in the body. It just goes around your digestive system and waits for its target. As the enemy bacteria attacks your body, our pyacins are needed for the rescue. The E. coli create custom-made pyacins with tails that matches the enemy bacteria. Those pyacins are being released, they bind to the enemy bacteria and perforate its membrane, and by that they're basically killing it. And once again, the day was saved by our pyacin. Not the hero we deserve, but the hero we will need in about 20 or 30 years. Okay, so let's have a short antimicrobial comparison. We've got our pyacins, we've got phage therapy, and we've got antibiotics. At the moment, they're all able to kill the enemy bacteria, but only phages and pyacins are specific and do not cause collateral damage to the body or the microbiome. It's really hard to find new antibiotics, but phages are all around us, and we can use the latter in order to create new pyacin systems, and we're going to talk about it in a minute, don't worry. Phages contain DNA and they have high mutation rate. That makes them a bit unpredictable and therefore a bit more risky. but vaccines and antibiotics do not carry DNA at all. And as phages destroy an enemy bacteria, that's called the release of toxins. Vaccines do not cause such thing, and some antibiotics do and some don't. And our last issue is the resistance mechanisms. Here you need to understand that biocides in nature are only being used against certain strains of Pseudomonas. That means that most of the bacteria in the world never met a biocide, never had to deal with it. This weapon is orthogonal to the other weapons that we talked about, like phages and antibiotics. And this is important because there are resistance mechanisms against antibiotics, and even against phages as CRISPR or restriction enzymes. Well, that was a futuristic dream, right? But we believe that in order to achieve fantastic things, you need to have a bit of a fantasy. So we've decided to create a proof of concept that will show you that biocides can be used as a future antimicrobial drug. This POC consists of 3 stages. The first is the bacterial target selection, then the system construction, and the last stage is the optimization of the system. So let's dive into the target selection. This is our biocide, you remember it, right? In nature, it's only being used against Pseudomonas, so how will we use it against other bacteria? That's a really good question. But luckily for us, we've got phages. And phages, some families of phages look really like our biocide. The main difference is only the it's full of DNA. And it's not only the presentation, even in real life they look the same, believe me. And what we can do is to take the tail of the phage and to use it as our new biocide tail. Another approach that is possible is to take only the tip of the tail and to create a fusion gene, partly made of the biocide tail and partly from the phage tail. Well, it looks great in the previous slide, but apparently it's not so easy in real life. And in order to find those genes of bacteriophages that can be used as our new biocide genes, there is a lot of work to be done, and we use bioinformatics method in order to do this. So we started with a huge database that consists about 500,000 bacteriophages genes, and we wanted to narrow it down and to find the tails genes. In order to do it, we've used 2 heuristics. The first one is pretty simple, we just look for genes that contains the word tail in their name. And the second one, we took the gene and we split it into short sequences and checked how many of those sequences we can find them on and also in the original biocide tail. After we have our tails candidates, we're looking for the tails that can be used as our new biocide tails. And this is important. It's not so easy because we need our new tail. On the one side, it needs to match the original base plate of the biocides, so they will bind. But on the other side, it needs to be different in order for it to bind to a new enemy bacteria. So for that, we created an alignment algorithm that checks especially for that. And the last stage is the optimization, where we optimize our parameters. In order to validate it, we took a tail that has been used in the past as a new biocide tail, and we got a really high score, so it really works, not just a slide. And we took this entire system and we inserted it into a software that we call Tailor Swift, because we find tails really swiftly, fun name, and a really beautiful logo. And what's so amazing about Tailor is that anybody can use it. You don't need any knowledge of the algorithms behind it. You can just enter the enemy bacteria and we will find the tails for you.
So, let's dive in a bit to the structure of our system. First, we would like to emphasize that our proteins are produced by a highly complex and large gene cluster. This cluster contains regulatory, lysis, and structural genes. We are talking about something like 20,000 base pairs. From this cluster, we identified 3 basic elements, which we divided into 3 different plasmids in order to control their expression. So, the first plasmid is the part N plasmid, which contains the natural activator for our pyroxenes, and is activated by an external inducer like vanillic acid. After the activator is produced, it activates our second plasmid, the cluster. The activator increases the transcription of the cluster, which is responsible for the body of the pyroxene. And now that we have the pyroxene's body, it leads us to the third and most important and interesting element in our system, the tail. We constructed 4 different tail plasmids, each one of them contains one tail and is activated by external inducer like IPTG. And that's it. Now we have pyroxene which is ready to attack. Let's look at some cloning results. Here we can see a pick and plate after transformation of the part N plasmid. We compared between the sequencing results to the wild type part N sequence, and it was 100% identical. In these 2 pictures, we show the cloning of the gene cluster into the E. coli by the Golden Gate reaction. In the upper plate, we see the existence of flux C in the desirable backbone, which gives the blue color to the colonies. In the lower figure, we see white colonies that represent the transformed bacteria that has the cluster instead of the flux C. In these 2 panels, we see successful induction of 2 tails that were produced by our engineered E. coli. So, the final experiment in our project was the ultimate blood assay test, where we tested our system on 3 different targets: the Salmonella, E. coli, and Pseudomonas. What we did in this experiment was to take the engineered E. coli, add the inducer, and drop all the lysate over the plates that already had the target bacteria. After this, we checked if the bacteria grew or not. On the right and in the middle, we see that there were no plaques in places we expected to see them. While this picture, the left one, showed not enough specific results, because we see plaques in places we expected to see them, but we also see plaques in places we did not expect to see. So further examination is required. But what's special about this experiment is that the Salmonella tail was found by our own software, the Taylor Swift, remember? We believe that with a few additional iterations, the specificity can be improved. As mentioned before, the pyroxene engineering is a very new topic, and the system had to be explored. So we would like, if any iGEM team, future iGEM team will continue our work, because the future looks promising.
After consulting with experts in the field, we understood that many projects like ours sometimes fail in the crucial step of upscaling, moving from small production to mass production. Therefore, we built a simulative model in order to understand better the interactions between pyroxene and bacteria. Later, based on this model, we built a pyroxene usage optimization algorithm in order to optimize the use of pyroxenes. And they are based on, the optimization is based on the model. So let's start with the model. This is a simple example of the model and what we can understand from it. The following video has the bacteria and the pyroxene populations over time. The right side is the bacteria amount over time, left is pyroxene amount over time. A few things we need to notice when we look at the video. On the right, we can see the bacteria amount is uniform in the entire 2-dimensional space. Pyroxenes, however, are located in 9 specific and even drops. There's a 20-minute delay time between the binding moment of pyroxene to bacteria until the pyroxenes actually kill the bacteria. So what we can see is that the pyroxenes drop and diffuse in space while the bacteria divide and rise, and there is no impact. Only until the delay time passes now, when we can see that the bacteria die because of the connection for the pyroxene. So the model was based on these equations. We based it on the Lotka-Volterra prey predator equations from ecology. And on the upper equation is the pyroxene amount over time, the lower is the bacteria amount over time. Both equations have 2 parts. The first part is the diffusion part. The second is the pyroxene bacteria interaction part. So these are the 2 equations. Later, we built our usage optimization model. So what we wanted to understand is how to use the Pythons in the best way possible. So the user inputs these parameters to the program, mainly the number of Python drops. All parameters go into our algorithm. And let's say we start with a uniform bacteria distribution like we saw in the video. And we wanted to locate three drops of Pythons on the 2D surface. What's the best way to locate them? That's the output of the algorithm that shows us that these locations are the best according to the parameters inserted by the user. That's the best way to locate the Pythons to kill the bacteria in the fastest time. So we showed you all three POCs. We showed you the target selection, the system construction, and the modeling and optimization. When we look to the future, we just showed you the Python, the medical implementations of Python, but the potential is giant, is incredible. So it can also be used for water purification or even agriculture. So the uses are infinite. And like every project in iGEM, nothing is complete without human practices, so Ophir will tell you all about it.
Thank you. So human practices is about involving your environment with your project, get some feedback, implement it, and improve. We have taken on two important goals. The first one is to find a novel way to fight antibiotic-resistant bacteria. And the second is to make science accessible to the public and raise the awareness of this global issue of antibiotic resistance. After consulting with experts, with Dr. Dean Scholl regarding working with the vaccines, and with Dr. Jonathan Lelouch about the in-body delivery of the vaccines and Israel's most common antibiotic-resistant bacteria, we implemented this expert's opinion into our project. During the year, we collaborated with a few iGEM teams from all over the world, starting with Instagram challenges to participating in a hair donation event, while the peak of our collaboration work was the mini jamboree that we hosted in Tel-Aviv University on October 6th. In this meetup, all the Israeli groups came, and even a special representative from iGEM team from the Netherlands. The aim of this meetup was to provide a platform for all the groups to present their work, get some feedbacks, and improve towards the giant jamboree. Throughout the year, we aimed to inspire the public about the potential of synthetic biology to solve world problems and the potential of our project to fight antibiotic-resistant bacteria. Furthermore, we believe in raising the awareness for this problem and pass the important message of right usage in antibiotics. For that purpose, we had a few lectures at various events and conferences during the year. We presented our project and the iGEM competition for the first time in the annually and largest Israel Biomedical Engineering Conference, which is called the ISMBE. In addition, we participated in a Science at the Bar Night, which involves giving scientific lectures in bar. 10,000 people arrived to the Israeli Researchers' Night that was held in Tel-Aviv University. During this event, we presented our project in a more friendly way so the children could understand. The fun part for the children was an experiment we did to extract DNA from strawberries so they could touch it and feel it. That was all for today. Thank you very much for listening.